comes up, I will share it with the class. Okay. Mm-hmm.
So we were allowed to turn a rough draft here?
No, they just showed it to me. You cannot turn in anything. They just have something in their note to me. I said, fine. Yeah. But don't turn in anything used, please. Anything that is used, don't, like, don't, no. I mean, use syringes or anything. Don't put anything on posters or anything that is used. That is just simply gross. That is just not right. Not right. Yeah. Pardon me? Next Thursday. Yeah. Mm-hmm. Yeah. Mm-hmm. Mm-hmm. Yeah. Yeah. And please, try to send me or a copy of your poster in a digital format. So if I want to reproduce it, okay, if I want to print it in a digital format, I could do that. Okay. All right. Yes, sir. Yeah. Yep. Yep. Oh, 12. Okay. Yeah. Oh. Okay. I will try. Sure. Oh, why am I doing that? Mm-hmm. Found the poster. Yeah. If you turn the lights off, please, all the lights, so that you can actually see it really nicely. This is about 8 or 9. I'll give it. Okay. This still needs a little bit of editing, more modification. She is working on it. All right. Thank you. Orlando, you can turn the lights back on, please. Thank you. Thanks. All right. No, she's a school teacher, high school teacher. She's working. She's going to be a high school teacher. She's in my class. Yeah.
That's on a board?
Hmm?
Is that on a board?
I'm not sure. Yeah, I didn't ask her. Yeah. Yeah. Alrighty. 13. Chapter 13, alrighty. First of all, let me go back and make the correction where I got stuck last time with the spikes. Okay. I made an error in saying all that have spikes, they cause hemagglutination. That was not right. I double checked and found out that not all viruses that have spikes can cause hemagglutination. Only certain types of viruses that have spikes can cause hemagglutination. And here are some of the examples of those viruses. Please go ahead and write down the names of these viruses. And HIV is not among these examples. Among the spikes, under the spikes, mumps, variola, vaccinia, and influenza viruses are some of the examples of the viruses that do cause heme agglutination, clumping of the blood. HIV does not. Correct. Yeah.
These are the ones that do. Right?
Where did I stop last time?
Complex viruses.
Structure of complex viruses?
On isolation and cultivation.
Oh, isolation and cultivation. Thank you.
What is variola virus?
Variola virus. I did not check up on what do they cause. Sorry. Yeah. Finish? All righty. Isolation. Did we start or no? No. Okay. Let's start with the isolation of bacterial virus. Bacterial viruses can be isolated by using two different methods. The first method is called suspension method, which is not very common. Okay. And the reason it is not common is it's kind of hard to remember the turbidity. If the turbidity was high or low at the beginning of the experiment and at the end of the experiment, unless you have that machine that is called spectrophotometer, which reads the turbidity, OD, optical density, then it's okay. But what if you don't have that machine, spectrophotometer? This is how this method works, suspension method. Suspension method for the isolation of bacteriophages or bacterial viruses. The best place to isolate bacteriophages is what? Sewage water, human sewage, because bacterial viruses, they infect, okay? They live in our intestine all the time. So if you go to a sewage water treatment plant, okay. Sample. That may have bacteriophages. First thing you want to do is filter it, okay, and get rid of the large debris and get your filtrate. Filtrate. Question? Go ahead. Sorry. Filtrate. And filtrate may have bacteriophages or phages. Now, in this explanation, I'm going to be making one mistake. Let's see if you can catch that mistake. Same mistake on the test. So that's going to be kind of your bonus question. After this, you are going to take your host in a bacterial culture medium. Bacterial culture medium. And add filtrate to this tube, bacterial culture medium. And then incubate this tube for at least 6 hours. Tell me, if bacteriophages are present in that sample, what will happen to the OD, optical density of this tube? Or go down? Go down, very good. It's going to go down. Turbidity, cloudiness, because when bacteria multiply, virus is going to infect them and kill them. So the turbidity is going to go down. Let's suppose, remember, if you don't have the machine that reads the optical density, it's going to be hard to remember. Is it more cloudy or less cloudy? That's why this method is not very common. Okay? But let's suppose we had the machine spectrophotometer. The initial optical density of this suspension was 0.50 at the beginning of the experiment. At the conclusion of 6 hours, the optical density went down to 0.02. If this happens, that tells you that the virus is present in that sample. On the other hand, if the optical density went up from 0.5 to 0.90, the bacteria, number of bacteria went up, cloud increased. Okay? Make sense?
So the virus is destroying, eating, and
Yep.
making it less cloudy.
Exactly. Virus infects the bacteria, multiplies inside it, boom, kills it. Every 20 to 30 minutes, virus goes in, and 250 to 300 new viruses come out, and they infect more bacteria. Okay? That's why you don't have to wait 24 hours for the results to see. In about 6 hours, 4 to 6 hours, you can see the results. Okay? Any problem with the results? Have you noticed anything wrong with this experiment, or we are okay so far? Yep.
What if the virus doesn't lyse the bacterial cell?
That's always a possibility. There are two possibilities. He is absolutely right. What if the bacteria doesn't kill? Absolutely. Yeah. When a virus, I was going to ask this question, when a virus infects a bacterium, it always kills the host cell. True or false? False. Okay, let me show you a diagram. Okay. When a virus infects a host cell, animal or bacteria, it always kills the host cell. False. Magic Johnson. God bless him, still alive, right? Okay. Here are the two possibilities right here. Virus goes in. This is what we are hoping to happen, what's happening in our tube. Goes in, starts to multiply, and kill the host cell. That will reduce the turbidity. Okay? Killing. So we are making an assumption that the Lytic cycle will take place. Not always. Sometime, no one knows, the viral DNA may become part of the host DNA. You see, pink DNA becomes part of purple DNA. This is what is happening in Magic Johnson. This is called lysogenic phase. There may not be any breakdown of the host. When the host cell reproduces, so does the virus. This is the carrier phase. So the original question, when a virus infects the host cell always dies. Not. Who determines which way the virus goes? Big boss. Actually, your immunity, the strain of the virus, therapy, and so on. But I strongly believe the big boss decides, really. Those are all the stories, really, here.
So the evolution, like that new flu that's in Japan, that they say made a jump from birds to humans.
New strains. New strains. That's why we don't have a good flu vaccine strain. They change their protein jacket so frequently. They don't have a good vaccine.
So it just happens that that protein jacket.
Exactly. You have vaccine. They make a guesstimate every year what type of virus you may have this year. So they take a guesstimate from the previous years and they just take 10, 15 capsids from the previous years and they throw it in a vaccine. So you have some years it may work, some years it may not work. That's what they do.
So it's just a calculated guess of what percentages.
That's it. That is it. Yep. Yep. All righty. Second method, which is more common, okay, because you can visually see the effect of the virus on the cell. That is called the plaque method. Plaque method. And plaque method is actually continuation of suspension method. Okay. So no problem here so far, no mistake. So you just missed one question on the test. Okay. Here's the mistake. Okay. I asked you, okay. So I say, Garrett, please go to the lab and get me bacterial culture medium. What would you get me? Broth. Broth without any bacteria. This tube that you have right here, it has no bacteria in it. So I need to cross out this word. Now I ask you, Garrett, please go to the lab and get me some bacterial culture. Now the tube that you're going to bring me has bacteria in it. That was the mistake. Just the medium, just the broth. Bacterial culture medium is just the broth. Now I send him and ask him, please go get me some bacterial culture. Now you have a tube with the bacteria in it. Okay, see the difference? Bacterial culture medium is a medium that is used to grow bacteria.
There should have been an inoculation.
Yep. You see, I should not have put the word medium here. This word should not have been here. So I had to erase that word. Now it is fine. You cannot grow virus in bacterial culture medium. You can grow virus in bacterial culture. Okay? Yeah. Now, let's talk about plaque method. In plaque method, which is continuation, instead of incubating this bacterial culture tube, you take a nutrient agar plate and you swab this suspension. You take a sterile swab and you swab this suspension on this plate. Now remember, what do we have in this tube? We have bacteria and we have viruses. All right. Now we incubate this tube for 6 hours. 6 hours, 37 degrees. Bacteria is going to grow. Virus is going to infect bacteria and kill it. What do you think is going to happen? Yep. Presumably, if lysis takes place, you will have clear spots on the plate. Wherever the virus is going to infect and kill the bacteria, you will have clear spots. And these clear spots on bacterial lawn, they are called plaques. Plaques. This method is more common because you can visually see the area of lysis. Animal viruses. 60, 70 years ago, choices were very limited to grow animal viruses. Scientists, they had to use animals, complete animal, whole animals. Okay. And animal rights activists, they were burning labs. They still do. Okay. But now scientists, they have more choices, better choices, like cell cultures. Okay. Cell cultures or tissue cultures, primary cell lines and continuous cell line. Embryonated eggs. Last year there was shortage of vaccines because they did not have enough embryonated eggs. This is the diagram I got from Google. Same diagram, similar diagram is in your textbook, but it doesn't tell you what type of virus can be injected, which part of the egg. Slightly better, but I will not test you on the name of the viruses, okay, just to give you an idea, okay. What's the difference between, you do need to know, what is the difference between primary cell line and continuous cell line? I will ask you that. What is a cell line? It's a tissue culture, okay. This diagram is in your book. Primary cell line. You are a scientist doing research on hepatitis virus, and you want to see the effect of hepatitis virus on the second generation of the cell. You do a biopsy of the liver, and you put the cells in a petri dish, okay, in a special medium. The medium that they use to grow animal cells is called Eagle's medium, okay. The disadvantage of most living cells is that they can only grow for 1 or 2 generations, and then they die, okay. What if you want to see the effect of hepatitis virus on the 5th or 6th generation? Can you do that? No, because this cell is going to die after 2nd generation. There was a lady in Virginia by the name of Henrietta Lakes, black lady, who had cervical cancer. She was in Johns Hopkins Hospital. Okay, she is the only known person who had cervical cancer, and her cervical cancer cell had this unique characteristic that her cell will not die in vitro. Her cells were placed in a petri dish. They will not die. Scientists had been trying to grow cells in petri dish forever. They have been trying to induce even cancer cells in a petri dish. They will die for a few generations. They will multiply for a few generations, and they will die. So scientists have been trying to make cells into Energizer bunny cells. No success. But Henrietta Lake's cervical cancer cells were the only cells that will never die. Those cells are called HeLa cells. HeLa, after her name. Okay. HeLa cells, Henrietta Lake cells. Those are abnormal cells that just never die. Literally, they never die. They keep on multiplying, multiplying, multiplying. Okay? Sure.
The other cancer cell, even though they are immortal.
Immortal, yep.
They will die. Eventually, they will go through apoptosis and die.
They will eventually die because of bacterial contamination and lack of nutrients. Yep. Yep.
This HeLa cell, even though if it's not replated to 10 out,
Yep.
it will just continue.
On top, transform. But the scientists, they have done this now. They take the gene, the MAD gene from the HeLa cells, and they can take that gene in any other healthy cell. Let's say they want to put this in liver cell. They can take that gene and put it in liver cell. Now they have a hybrid cell of liver that will keep on multiplying. Yeah. So this industry, science industry, trillions of dollars on her behalf, and her family is getting zero. Nothing. Go Google it. Google it. There are documentaries on her. I know. She is the only known person on this planet. Her cells are doing this. And her cells are being used in academia. We use it in our in lab. Henrietta Lake cells, okay, HeLa cells. Research industries, pharmaceutical industries, they are using for research purposes, but her family gets zero. I'm sure they did and all that,
So the
but
genetics from her cells tie it into any other cell?
Just keep on multiplying. That's called continuous cell line. They literally never die. Yep.
How long ago did this happen?
I think it's 1960s it happened. And they were actually stolen without her knowledge. She did not give consent. Yeah, she never gave consent. They were stolen.
They just did something
Yeah.
and realized
Yeah.
this is.
Yeah. So, yeah. All right, welcome to America. All right. Yeah, all right. Plant viruses. Plant viruses, remember Dmitry Ivanovsky? Plant viruses, if you're a plant biologist, you can use whole plant like Dmitry Ivanovsky did. He was working on tobacco plants. You can do that. But nowadays, we do have plant cell lines.
Sorry, Professor, sorry. You explained cell line is tissue culture. What's primary?
Primary cell line grows for only 1 or 2 generations. They multiply for 1 or 2 generations and then they die. Continuous cell line is a hybrid cell. Normal cell and a cancerous cell like HeLa cells. Hybrid between 2 cells. They never die. Continuous. They keep on multiplying forever. So plant cells, there are a few plant cell lines available so you can grow plant viruses in a petri dish too. Multiplication of viruses. We will first look at the multiplication of bacterial viruses, then we will look at the multiplication of animal viruses, especially multiplication of HIV. Okay. Bacteriophages involves 5 major steps: attachment or adsorption, adsorption and absorption. Everybody okay? Okay. Penetration or entry, biosynthesis of viral components, maturation or assembly, and finally release. Here's a diagram. Adsorption. Not every bacterial virus can infect any bacteria. There are special bacteriophages that infect special bacteria. A virus that infects E. coli cannot infect Salmonella. And special bacteriophages infect especially E. coli. Okay, very specific. First step, adsorption. The virus attaches to the special receptors on the surface of the bacteria. Then penetration. Not the whole virus penetrates. Why not? Because of the thick cell wall. Cell wall stops the penetration of the virus. Only and only DNA penetrates. How? You see this little tail-like structure? Inside this tail, they have this needle right here, which actually the tail shrinks, and this needle actually penetrates through the cell wall, and the nucleic acid is injected into the bacteria. Very sophisticated process, actually. So, and the body of the virus is left outside. Okay? And after that, there is, Sandy, could you please put your phone on my table?
I've got an emergency going on.
Outside. Biosynthesis of viral component. Okay. All the parts are made. All the parts of the viruses are made. And then maturation or assembly. Okay? As you can see right here, just like a car, the parts are made, okay, and then all these parts, and this is when this is when the mistakes are made. Like in transduction, remember, some viruses are they pick up the bacterial DNA, right? This is when during transduction, okay, some viruses may pick up the DNA of the bacteria. And then finally, release. Okay. There are three terminologies that are associated with the process of replication. First is called eclipse time. Eclipse period, actually. Eclipse means what? Eclipse? Eclipse? Eclipse of the moon, eclipse of the sun. Block, to hide. Eclipse of the sun, partial eclipse, full eclipse, to mean disappear. Partial eclipse, right? All right. Okay. Eclipse means when no complete viruses are detectable in the host cell. That is eclipse period. So eclipse period, a period in which no complete viruses are found in the host cell. So if we go back, sorry. Okay, sorry. How about the first phase? Which of the 5 steps do you think should be included in the eclipse period? Okay. First period, eclipse period or not? Yes, there are no complete viruses. How about the second phase? Eclipse period? Yes. Third period? All the parts are made, but there are no mature viruses, right? Okay. So the first 3 periods are called eclipse period. What do they call Mitsubishi eclipse? Really? Mitsubishi, the car? Really? Why did they name it eclipse? Because it is so fast it disappears. Really? I think so. Really? It leaves you in the dust, I think. Yeah, I think. Yeah. Okay. All right. Yeah.
Is that why, let's say a nurse gets a needle stick, they
Yeah.
get tested for HIV and they get tested again in 2 weeks and then in 6 weeks because it could be the eclipse period?
Very good. Very good.
It's
Yeah.
not detectable.
Very good. Very good. Yeah. Yeah. Burst time. Burst time. Burst time is the time it takes, is between the attachment and release. The time it takes for the virus to attach and release, or the time it takes virus to complete its cycle. And it's about 20 to 30 minutes from time, burst time from attachment to release. Burst size, okay, fine. Number of mature viruses released from a single host cell, 250 to 300. Burst time between attachment and release, 20 to 30 minutes. All right. What is next? Multiplication of animal viruses. I have picked HIV. There could be DNA viruses, there could be RNA viruses, but I have picked RNA viruses, which is slightly complicated. Okay, DNA viruses is straightforward. Most of the steps are same. One is slightly different. Attachment or adsorption, same. Penetration, different. Why do you think it is different? We are going to look at the diagram. No, very good. No cell wall, so the whole virus enters. Yeah. We don't have any cell wall, so the whole virus enters, penetrate. Uncoding, the virus literally strips. Once it is inside the host cell, it takes off everything. It takes off cells. Really? Just imagine stripping going on inside the cell. God. I wonder if there's any music going on inside the cell. Biosynthesis. And finally, maturation and release. Let's look at the diagram. This is HIV. All right. The first two steps, I don't know why couldn't they put them separately. They have put them together. Okay. Adsorption and penetration, they have merged them together. I don't know why. Okay. Adsorption. Okay. Fawzia just gave me this article, so I'll just make it a little bit more specific for you. P24 receptors. Yeah, these receptors that you see spikes, they are called P24 and GP41. If you are in, we were talking about spikes last time. Okay, so these little spikes that you see right there, they are GP41 and P24. Okay, so they attach to the receptors. Okay. And then penetration. The whole virus enters into the T4 cell. Okay. And then, this is bad diagram. In the older edition, they had a pretty good diagram where they actually showed the stripping. But here, this uncoating, but where is uncoating really? Where are the parts of the capsid? Where are the parts of the envelope? They are not showing any. But this is uncoating, fine. Okay, yeah. Now, the reason it's called retrovirus is this right here. Retro means backward virus. Normal cells, they use DNA to make RNA, right? But this virus uses RNA to make DNA. That's why this HIV is called retrovirus. Okay? Normal cells, they use RNA polymerase to make DNA. This one uses reverse transcriptase. Not going to test you on that, okay? Just the steps, nothing on from this. So RNA to DNA. Once the DNA is made, it becomes part of the T4 cell. Remember this term, please, right here. What's a provirus? When the animal virus becomes part of the host DNA, that is a provirus. Remember this term, please. Okay. When the animal virus, not necessarily HIV, when any animal virus becomes part of the host DNA, that's a provirus.
So when the sequence of the virus is included in the DNA.
Exactly. Yep. Now, if this is happening in bacteria, what would you call it? Prophage. Prophage. Not provirus, but prophage. P-H-A-G-E. Prophage. Okay. Now, here, remember from here, there are two possibilities. In Magic Johnson, the process has stopped right here. Okay. After this, no one knows why some people don't show signs and symptoms. Okay. After this, they become carriers. Okay. There is no replication of the virus. And the process stops right here. They become carriers. And some people, they start replication and they will show signs and symptoms. Some people, it's right here. They are carriers right here. Okay?
Because of the treatment?
No one knows, really. No one knows. Some people, they will start, they start to show signs and symptoms after 5-6 years.
Is it true that a very low percentage of population, I don't know how much, that they are naturally resistant?
There are, like this child, this kid, 2-year-old kid, or I think in Atlanta or somewhere, this kid was treated for 2 years, extensive treatment, disappeared, completely disappeared. They can't even find. Yeah, so different strains, different treatment, and really no one knows what triggers this. Well, yeah. But let's say something triggers this virus and it starts to multiply, biosynthesis of viral components up to here, and then maturation. Look what happens here. Where is the envelope coming from?
From the host cell.
Exactly. That's why sometimes your body has hard time identifying or responding to HIV, because the envelope is actually part of your own T4 cell. Look, right there. Okay? And then release. Okay? So adsorption, penetration, uncoating, okay? Provirus, biosynthesis, maturation, assembly, and release. Okay. Lytic cycle and lysogenic cycle. We already talked about this, but let's go over that again. Lytic cycle is a cell in which virus or phage will cause the death of the cell. Pardon me? Most of the time, but we don't know what causes lysogenic, what causes lytic. Yeah, but most of the time if a virus goes inside the cell, most of the time lytic cycle will happen.
Does it happen with HIV mass? Is the cell lyse when it leaves? When the virus
Yes, if the virus leaves, definitely the cell will die. Yes. Yep. So when the virus, some viruses, they may not cause the death. Instead, they may become part of the host. That is called lysogeny. If the cell has incorporated, if the virus has incorporated its DNA into the host, the same virus cannot reinfect the same host. They become immune, just like one sperm has fertilized an egg. No more sperms can fertilize the same egg. Okay, same principle. A lysogenic cell becomes immune to the reinfection by the same phage. And what's the advantage of lysogeny? Cells gain new characteristics. Okay. Lytic, lysogenic. May happen in bacteria. Same thing may happen in humans. Do viruses cause cancer?
Yes.
Yes, they do. Absolutely. Okay. We all have, when I say we all, plants, do plants have cancer genes? Yes, they do. Okay. Mm-hmm.
What would be a virus that would create cause cancer?
Pardon me?
What would be a virus that could cause
Papilloma, herpes, HIV. Yeah, I have examples right here. Yeah, a few. Let me explain first what happens here. Normal living cells all of us, we have, here's a normal cell, nucleus, DNA, different genes right here, X, Y, Z, A, B. Okay. Let's say this is gene X, and gene X has this little switch here. Just like this room has this light switches right there. Okay. You need those light switches to control the light in this room. Okay. This gene X makes a protein. This is called protein kinase. This protein kinase is extremely important for the regulation of cell growth. It tells the cell when to stop growing. It regulates cell growth. Okay. This little switch right here, it is normally off. Because of the presence of that little switch, this gene X is called oncogene, proto-oncogene. No other genes, they have that switch, but that gene has that switch. As long as that gene, that switch is off, this gene will functioning normally, keep on functioning normally, and keep on producing normal quantities of protein kinase. Now, all of a sudden, this person decides, or this person is exposed to asbestos, or ethylene oxide, or papillomavirus, herpes virus. Okay? This gene is now turned on. This proto-oncogene is no more proto. It is oncogene now. What is oncogene? Cancer gene. Okay? This gene will keep on producing abnormal quantities of protein kinase. The cell has lost its ability to control its growth. Cancer gene. Okay. All nucleated animal and plant cells, they have proto-oncogenes which produce normal substances. Excuse me. Okay. Normal functioning proto-oncogenes. Okay. 20 have been so far have been identified in animal cells. What can trigger them? Any mutagenic agent, radiation, chemicals, and viruses. Smoking and other chemicals that I talked about, radiation, and viruses. We are talking about virus, pox virus, papilloma, herpes, and HIV. Here are the examples. Last topic of this chapter, prions. I give you a brain of a cow from a mad cow disease and ask you to please take it to micro lab and grow some prions. How would you grow them?
In the living cell?
However you decide to. I just give you a brain in a liquid nitrogen tank and ask you to please grow it. Take it to micro lab, grow it.
Oh, you cannot grow it.
You cannot grow it because excuse me, prions are particles of proteins excuse me. Viruses, at least, in order to grow something, what do you need? You need a blueprint. When you want to make something, you want to build a home, you need a drawing of the house, how to build, what to build, right? What to make. Okay. You want to make a simple protein, like prion. You want to make you need to know which amino acid to put where, right? In order to make a protein, right? When you want to make a protein, the information for that protein comes from where? DNA, right? Where's the DNA here? No DNA. Where's the RNA there? No RNA, no DNA. There is no blueprint. There is no information. So you cannot grow prions. You cannot construct. They are just, where do they come from? Prions. Aliens. Huh? Huh? Yeah, most scientists believe that the host make his or her own abnormal proteins. Yeah, they're part of the disease actually.
That's where it goes back to what we were talking about Alzheimer's.
Exactly, yeah. They have been diagnosed or they have been detected in the early stages of Alzheimer's. Yeah.
So which came first, the mad cow disease or the prions?
That's a good question. They have not been able to establish that fact at all. Yeah. Fausia.
My question was that the prion is not the sole cause of Alzheimer's, right?
No, 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 not at all, not at all. They have not even established the link, strong link yet. Yeah, but they have found, yeah, prions in those patients. Yeah.
You can, so obviously you can test your prions. Can you have them, could it be a precursor, could be a marker for a disease without actually showing symptoms of the disease?
Too early to say that.
Way into the disease, or even dead to show the
They have found these in many, I mean, there are several, not many, several diseases, but they don't know. I mean, actually, I mean, what is the actual cause that leads to the formation of this? No. Definitely they have found in these diseases, mad cow, definitely. Incubation period for this in Creutzfeldt-Jakob disease, 15 years.
So we could just be living long and basically this is coming up.
Yeah, I don't know. I mean, Creutzfeldt-Jakob disease, not necessarily. There is no Alzheimer's. Patients do not have Alzheimer's when they get Creutzfeldt-Jakob disease. There is no Alzheimer's there. By the time they die, their brain has turned into holy brain.
Is that
I'm
the
sorry,
disease-
no fun.
The is that the disease happening in some tribes eating monkeys?
There's different schools of thought. Yes, people think, scientists think that this is how it was transferred from monkeys to humans.
But anyway, about the mad cow disease, so if 50% whose body is not making prion and eating the contaminated cow meat,
Yes.
to get the prion from the cow.
Yes, sure. That's why they burned hundreds and thousands of cows in the United States. That's not United States, United Kingdom, I mean. Yep, true. But people can make their own. Yeah, and your brain will be holy. Yeah, really, literally holy brain. You see the holes right there? These black spots are prions, and your brain turns holy. Look at how simple the actually transformation is. This is your normal protein in the brain. And this tertiary structure right here, tertiary structure turns into linear structure. That's it, that's prion. And this disease, there was I think on 2020, they showed a case of this gentleman from Russia who had this disease right here. He was alive at that time. Fatal familial insomnia. Person cannot sleep. Ever. Ever. Fatal. You die. You just abnormal, huh? You just cannot sleep.
So they just exhaustion or brain
That's it.
Maybe
Yeah.
Michael Jackson was getting that because
No, he
he
was.
wasn't, I don't know how to read this, he wasn't able to sleep in the.
That was exhaustion or something. Yeah, yeah. That's a different story. Yeah, that's a different. Yeah, 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 yeah. So you should be thankful for every morning we wake up that we can go to sleep. Really.
When that person got that, how long did they last before?
Few months, maybe a year or so. Google it and you will see his picture. When I was preparing this presentation, so I saw his picture. He was on Google. And there's a video too. Where you will see his yeah.
Oh, that comes from prion also?
In the brain. Yeah. He was in his 40s or 50s maybe. 40s, 50s. The people, they make their own prions. Yeah. Yeah. He probably ate or something? Nah. Unless there was some, I don't know. He contaminated beef or something? I don't know. I don't think he was eating monkeys? I don't know. So. Okay, we are going to continue with chapter 13, 14? Yes. All righty. 15 minutes? Yeah. I thought I'm finished. So. Okay. All right. 14, 15. 14, 15 is pretty straightforward. Late night. Late night, sorry. Sorry. So, normal flora and microbial diseases, just basic terminologies. Okay. Relationship between humans and microorganism. Okay. Let's go over some of the terminologies first. Pathogen. A pathogen is a microorganism that always causes disease. True or false? False. Pathogen, how many of you have pathogens? Everybody. Everybody. The reason you are not sick, because you're immune, your defenses are strong. Everyone has pathogen. Okay. But a pathogen will cause disease in a susceptible host. Give me an example of susceptibility, please. That is too extreme. I just might want a little susceptibility. That is too much. Scratch on the skin. Susceptible. Right? Scratch skin. Susceptibility. All right. Susceptible host. Scratch skin. Pardon me? Opportunist? Yeah. Yeah. You know, opportunity. Yeah. Whenever the opportunity is there, you ladies, I'm sorry, but it's not a very good example. Ladies using too much douche, right? Susceptible? Yeah, sure. All right. Pathology. Okay. Study of disease, scientific study of disease. All right. Disease. Dis-ease, away from comfort. All diseases are caused by microorganism. Not. Okay, some diseases are caused by taking my exam. Oh, God.
Lack of sleep.
Okay. Two types of diseases, infectious and non-infectious. Infectious diseases, where microorganisms are involved. Non-infectious, no microorganisms are involved. Infectious diseases could be communicable, right? Not all diseases are communicable or transmissible. Can be transferred from one person to another. Okay? Yeah. Infection. Simply, pathogen is present, but no sign and symptom. Infection. Okay. But when you can see sign and symptom, then you are diseased. Normal flora, normal microbiota. How about normal fauna of human? Fauna. No? Humans don't have fauna. Plants. No. Sorry. So flora, organisms that colonize normal human beings. Over 100 different species. Okay. Now this part I'm going to skip. You don't skip. You need to know this, but these are just names. Okay. So I'm not going to read all the names. There are different parts. Okay. Organs, different organs, and some of the examples. For example, skin. Here are some of the names of bacteria. Okay, that are found in the skin. Okay. There may be one question on the exam. If you want to do selective study, you can skip them. Okay? If you don't want to memorize them, it's your choice. Bacteria that are found in eyes, nose, throat, mouth. So I'm not going to read all those. I will go to the origin. Yeah, origin. I'll start with the origin. Yep, there we go. Oh, Which part of the body are sterile? BBB. Yep, blood, brain, bladder. Okay. Healthy fetus is sterile. The first contact of the fetus with the germ is when the birth membranes break and the baby comes out, and then the baby is exposed to the flora of the mom's breast and the skin and the handlers that handle the baby, the air that the baby breathes. Okay? Sure.
Say the bladder is sterile. Would that make urine sterile?
No, no, because urine has to pass through the urethra, and urethra is not sterile. Yep.
With catheter, it's sterile.
Catheter is supposed to be sterile.
But I have a question about the blood. How can we say blood is completely sterile? Because when we get any type of infection, any bacteria or viruses, doesn't mean necessarily cause the disease. The first place that it enters is the blood, through this, whether it's through the skin, through the mouth. Once it gets into the serum, it gets through the blood, and then it's the blood where the white blood cell fight infection. I mean,
In a healthy
we had
person,
infections, but can we
in a healthy person, blood is sterile,
Okay.
right? In a healthy person, it is sterile.
Well, it could be here, and the immune cells could be attacking and killing, but it has to get in the blood in order for the white blood cell to attack and kill it in a healthy person. So if we say blood is 100% sterile, that means it never get exposed to the microorganisms.
It is constantly exposed to microorganisms, constantly, because you have abrasions in your intestine, in your skin, and all that. So, yeah, I'll say 99.9999999%. Okay? All right.
So unless you're sick, it's
Sterile. Yeah. All right. Is that sterile enough? Okay. All right. All right. HIV, it can pass through the placenta. Okay. And there are many diseases that can be transferred at the time of the birth. If mom has gonorrhea, syphilis, chlamydia, they don't do vaginal birth, C-section. If you,
Sorry.
sure, go ahead.
Curiosity.
It's all right.
The streptococcus causes sore throat.
Yes,
Then through the blood, it goes to the heart and causes endocarditis.
absolutely.
So if the blood is sterile
Yeah.
all the time, 99.99, which is 100%,
Yeah.
how could it take this streptococcus and causes autoimmune disease or arthritis?
Oh, once you have strep throat and it goes to your blood, then it is not sterile.
So, but if we say it's already sterile.
No, of course.
It's not a closed system. It can't, the brain, the bladder, all that can become infected, but
Of
under
course,
normal
yeah.
conditions,
Yeah. It cannot be sterile all the time. That's, yeah, it cannot be sterile all the time. Yeah. Yeah. Yes.
And you know, here it says HIV cannot pass through placenta unless internal injury.
What does it say here?
But there it says differently.
What does it say here? What does it say here? Sorry, I changed my notes. Yeah, in my old notes, yeah, it does say that, Jackie. In old notes, it does say cannot, but yeah, they are changed. Yeah, please, yeah. Old notes, it does say cannot. Yeah. Yeah. All right. If you want to see some really, really horrific pictures, see gonorrhea that has passed through the membranes, birth membranes, and killed the fetus. Really, that's nasty. Relationship between normal flora and host. 3 types of relationship can exist between normal flora and the host. Could be positive, could be negative, or could be neutral. How much time I have? Okay, let's go. Yeah. All righty. Have a good weekend. Oh, one more thing I forgot. Extra credit for lab. David, you can go if you want to. No, okay, right? For lab, for 5 extra points, pick one lab, okay, one lab, and rewrite the principle only. You don't have to rewrite the whole lab. Rewrite the principle.
You said it was going to replace it. You had originally said you would drop the level quiz grade.
I lied.
We have it on.
Okay, if you want me to replace your quiz grade, that is how many points? 20 points? Do the whole darn thing. Okay.
We have
20 points versus 5 points? Okay, that's a lot. If you have to redo the whole thing, that's a lot. So your choice, you want to do the whole thing, 20 points, just the principal, 5 points. Your choice, okay? One is due by the end of the term, okay?
Okay, we have the diagram there, and then we have that. This is the pipe, and then we have like little ants in here, and we write the name of the bacteria and the organism, and then we write the disease that it causes.
leaving because the ants are wild.
Oh, okay. All right. Nice. Okay, very good. I apologize the other day.
I
You
was
didn't.
late.
You were, but I had doctor's appointment, so I'm so sorry. Yeah. Sorry. Yeah. Okay. Sorry.
I have a question.
Sure.
Reading this book, and it's an old book, and they're doing a safari into Africa, and the guy gets a cut on his leg, and the tribesmen were they said to use urine to help sterilize the cut.